My lab is interested in plants, lipids. We have several projects going on in the lab. Today, I want to share with you one of our most exciting story, at least I believe. It's about how we engineer plant leaf to accumulate oil. So in today's talk, first I will give a brief introduction to plant lipids and the methods we use to analyzing lipids. Then I will try to explain why we want to produce oil in leaves. This third part of my talk, I will try to describe the genetic strategies we use to increase oil accumulation in leaf. In the last part of the talk, I will give some thoughts on what we are going to do moving forward. So lipids are broadly defined as a group of hydrophobic molecules, and the majority of lipids are derived from fatty acids. So what is a fatty acid? Fatty acids are group of carboxylic acid with non-hydrocarbon chains. Here is chemical structure. The structure here is a 3D molecular structure of fatty acids. So the one for major class of lipids are glycolipids, include oil and membrane lipids. So oil, also called triacylglycerol, TAG or TAG in short. So as the name implies, triacylglycerol contains 3 fatty acids attached to a glycol backbone, so called triacylglycerol. So again, this is chemical structure. Here is a 3-dimensional molecular structure of triacylglycerol. So another similar compound called diacylglycerol contain 2 fatty acids attached to a glycol backbone, or I will call it DAG in short. So the DAG contains an empty position here. So where is the oil made? Shown here is a drawing of plant cells. It has rigid cell wall enclosed, and inside of plant cells, there are large number of cellular organelles, and here is a chloroplast, and endoplasm reticulum, or often called ER, a central vacuole, and among many others. So where is the oil made? In all the eukaryotes, human, plants, as well as yeast, ER is the major site of oil synthesis. So oil is synthesized in the ER, but they are stored in an organelle called oil droplets. Oil droplets can be visualized under electron microscope. Here showing the electron micrograph of oil droplets in the leaf cells. Oil droplets can also be seen using a fluorescence microscope. Here, leaf tissue first used to stain with fluorescence dye we call NARED. Here is how oil droplets look like under fluorescence microscope. So what is the membrane lipids? Membrane lipids is the building block of biological membranes. And the majority of membrane lipids are glycolipids with a hydrophilic halogroup and 2 hydrophobic fatty acid tails. And this amphiphilic property make this type of membrane lipids bilayer forming. in an aqueous environment. So in a typical biolayer, so the head group is facing outward, and the two fatty acids tail is facing each other, which form the core structure of biological membrane. So membrane lipids are often defined by the nature of a halo group. If halo group is the galactose, we call galactolipids. If halo group contains a phosphate group, we call phospholipids. And galactolipids, most of galactolipids are concentrated in the organelle we call chloroplast. And in fact, as much as 80% of chloroplast lipids are galactolipids. So inside the chloroplast, there are extensive membrane systems sitting in here. This membrane system we call thylakoid. So what's a thylakoid? Thylakoid is a cellular photosynthesis, a process by which plants use sunlight as the energy source to convert carbon dioxide into sugar with release of oxygen. So where membrane lipids are made, in plant cells, there are two important organelles that coordinate and synthesize the majority of membrane lipids. These two organelles are chloroplast and ER. So how we normally analyze lipids? Lipids can be accurately quantified by using sophisticated machines such as GCMS, LCMS, and more. So a simple way of looking at the lipids is by using a thin-air chromatography technique. So here I show you an example how we use this TLC system to separate oil from other lipids. Basic procedure include extract lipids from, for example, here leaf with organic solvent, then spot lipids on the end of TLC plates. The TLC normally coated with silica gel. Then we place this TLC in a solvent system, which allow the carrier lipids moving upward to separate lipids, different lipids. So after separation, the plates are stained with a reagent. Here I show you the chromatogram, and here is oil. And often here we also can see that this the lipid intensity, this band intensity and size often can give you first indication as to how much lipids is present in the lipids extract. In this case, we see much more lipids in this sample than in these samples. So the second part of the talk, I'll try to explain why we want to produce oil in leaf. So our society become dependent on fossil fuel for energy. And those fossil fuels are formed hundreds of millions of years ago, and they are not renewable, and they will eventually run out. In addition, due to the consumption of fossil fuel, carbon dioxide pollutants are released into atmosphere, which cause major environmental concern. So perhaps one of the greatest challenges facing mankind is to develop alternative, clean, renewable energy source to replace fossil fuel. So biodiesel derived from plant oil is such an alternative because biodiesel is renewable and as well as carbon neutral. As shown here, biodiesel is made from plant oil. So carbon dioxide released during biodiesel combustion can be refixed by photosynthesis. So into sugar. Sugar can be converted into carbon dioxide oil by enzymatic machinery present in the plant cells. This whole process is sustainable, and fixed energy from sun use this energy to drive cars, for example. So what is biodiesel? Diesel is, consists of fatty acid esters. It's made by reacting methanol typically. Okay, we can use other alcohol as well. So typically by reacting methanol with oil to produce fatty acid methyl ester in this case, by reaction called transesterification. So in terms of chemical property or energy content, biodiesel is very similar to fossil fuels. And in addition, biodiesel is clean and have no emission and is compatible with existing fuel technique and infrastructure. Due to this and many other advantages. So the use of plant oil for biodiesel production increased rapidly in the world, include US, Europe, Union, and other parts of the world. So right now, most biodiesel is made from seed oil, mainly canola seeds and soybean seeds. So soybean and canola are also the major oil crop in the world, and with oil palm and sunflower. So right now, our annual plant seed oil production is around 160 million tons a year, and most of this are usable for food. I mean, right now, our fossil fuel consumption is 4,000 million tons per year. And so even we use all this plant oil to make biodiesel, it only can replace a small fraction of fossil fuel usage. So for biodiesel to make an impact on the fossil fuel consumption, we needed to produce more plant oil. So one way solution to expand oil production is produce oil in vegetative tissues of traditional crops such as corn and soybean. So compared with producing oil in seeds, producing oil in vegetative tissue has at least two advantages. First one is biomass of vegetative tissues much higher than seeds, right? You can see that seeds only make a small fraction of total biomass. And further, if you produce oil in a vegetative tissue, this portion does not compete with food production. So another way we can expand oil production is to produce oil in a bioenergy crop, which normally has very high biomass yield. So, however, leaves or vegetative tissues, they carry out photosynthesis, produce sugar, starch, instead of oil. So as shown here is a cross-section of a mature leaf. You can see that in most of the cells, chloroplast is the main organelle here. We can see under this magnification. So chloroplast, as I mentioned before, is an organelle that's responsible for photosynthesis. So if you look at a higher magnification, you see that the chloroplast is filled with starch. We don't see any oil. So in fact, if we use GCMS or other machine, the oil is immaturely for Y-type plant here. It's normally lower than 0.05% by dry weight. Most of the lipids in the leaf tissue is membrane lipids, constitute as components of photosynthetic membrane and other membranes. So in the next part of the talk, I will try to describe the genetic strategy we use to increase oil accumulation in leaves. So I would like to point out that most of our studies use a plant we call Arabidopsis. This is a model system for plant research, including lipids research. And the reason we use Arabidopsis is because this plant has a short life cycle, 3 months from seed germination to seeds harvest. And it has a completely sequenced genome and a large collection of mutants, including lipids mutant. Most every gene that is involved in lipids metabolism already has, we can find a mutant, carry mutation in almost every gene that involves lipids metabolism. And we can, this mutant, some of mutant are called lockout because the whole gene is removed or disrupted. Or we have mutation. For example, they have one nuclear mutation which disrupted the gene function. And a lot of other mutations, mutant as well. And this plant is very easy to introduce a foreign gene. In other words, it's very easy to do genetic transformation. You can steep the flower in the solution, then very rapidly, very simply, you can introduce the foreign gene. We can do overexpression, for example. Overexpression here, I want to mention that overexpression is a technique to increase the expression of a specific gene level or expression of protein level. And also very easy to grow and handle. They can grow happily in the agar plates and also on soil. And in this presentation, I will try to follow the nomenclature adopted by Arabidopsis research community. So I will use the capital letter without italics to indicate the proteins. For example, TGG, this is the most important protein, as you will know in my following talk. Or mutant, I will use the italicized lowercase for here is a TGG1 mutant. Remember this TGG1, I will show this is the main target I will want to mention many times in the following talk. So, as I mentioned before, there are two organelles, chloroplast and ER, which are important in a lipid synthesis pathway. So, fatty acid is a precursor for synthesize both membrane lipids and also oil are made almost exclusively in the chloroplast. And polyfatty acid, about half, is used inside the chloroplast to make glycolipids. A lot of polyfatty acid are exported into a lot of organelle, ER, or we call it endoplasmic reticulum, to make phospholipids. So polyphospholipids made in ER, they re-import into chloroplast to support glycolipids synthesis. Here we have a protein called TGG1 is responsible for transport phospholipids back into chloroplast. In a normal condition, as I mentioned before, in a wild-type leaves, very little fatty acids made used for tag synthesis. Even though these two tag and membrane lipids, phospholipids and tag synthesis occur at the same subcellular compartment in the ER, and the lipids oil is very low. So the first strategy, we know that fatty acids common precursor for both membrane lipids as a halo group, make membrane lipids. And as well as tag, I show it's oil droplets, this is tag synthesized. The fatty acid is same precursor. These two pathways compete for the fatty acid. And we know in many studies in both plants, yeast, and mammalian system that member lipid level do not change much because they have a homeostasis mechanism. So this homeostasis, in other words, homeostasis to maintain the member lipids almost constant and many, many different conditions. So this means that the demand of fatty acid for member lipids synthesized won't change too much, doesn't change too much. So we figure out that one way, if we want to increase oil-based synthesis, we can increase the availability of substrate fatty acid. In this way, we can increase the fatty acid synthesis to produce more fatty acid per cluster, pushes this thing towards oil-based synthesis. So when I was Michigan State, we discovered a type of mutant we call a type of mutant that are defect in this pathway, mean transfer phospholipids back into chloroplast. And as I mentioned before, this pathway is mediated by a protein called TGD1 protein. So by extensive genetic analysis, we spent at least 3 years or something to figure out, at that time, in 2003, the full sequence genome is not yet available. So we spent a lot of effort to figure out what is the mutation responsible for this phenotype. Eventually, we figured out that this mutant carrier one nucleotide mutation in a gene named TGD1. As I mentioned before, it's responsible for transport lipids from ER back to the chloroplast. So this mutant we named the TGD1 mutant, and here's how TGD1 plants look like. It's look fairly normal, I would say, except that the growth is slightly reduced compared with the Y-type normal plants. So by use biochemical approach, here we use radio labeling. We use carbon-14 labeled acetate. This acetate is a precursor for fatty acid synthesis. So if we feed this precursor, we can choose how much this fatty acid is going this different pathway. And by doing this, we found that this TGD mutant rate of fatty acid synthesis, they produce is increased almost fourfold compared with Y-type. So they produce almost fourfold more fatty acid compared with Y-type. So this where this fatty acid goes, it goes to glycolipid synthesized in the corpus. And it also goes to here, phospholipids and endoplasmic reticulum. And importantly, there goes a lot of fatty acid goes to oil because this pathway is blocked. So here is how this TGD mutant also illustrates how this homeostasis mechanism take place in the plants. So when we look at the member lipids content, they do not change. I mean, the same level, TGD1 and Y-type, member lipids content is exactly the same. This is what happened. We have fourfold increase in the fatty acid synthesis, and more fatty acid goes into here to make a glycolipids by chloroplast itself. So even though this pathway is blocked, they still can make same amount of glycolipids. So when you look at the oil content, we found that tenfold increase in the leaf. Of course, I always talk, everything is leaf. The tenfold increase in the oil content in this mutant compared with white to 0.5%, something like that. So even though we have fourfold increase in the rate of fatty acid synthesis, we produce fourfold substrate, and this pathway is not changing dramatically. This pathway member lipids is maintained, remain constant, and we still can get this much of oil. So we figure out that maybe the pathway that convert fatty acid to TAC here, this step might be limiting. So we know that in studying seeds, showing that there are two enzymes. One is called PDAT, one is called DGAT. They are important in convert fatty acid into TAC. Specifically, both these enzymes are responsible for attach larger fatty acid to the empty position of diisoglycerol to make it triisoglycerol. So to test whether they play any role in oil biosynthesis in leaf, we use transgenic approach. We'll overexpress this PDAT1 or DGAT1 in TGD1 mutant. We see whether this two enzyme is limiting. Overexpress, we increase the protein as well as membrane level for these two enzymes. So as shown here, if we overexpress DGAT, we don't see much difference. Oil content here is oil TAC oil content compared with the TGD mutant. And but if we overexpress PDAT, we see 50-fold increase. So this data suggests that this enzyme is very important, the key player in oil biosynthesis in leaf, at least in Arabidopsis, but it definitely other system probably is different. So here's how oil droplets look like in this TGD1 overexpressed PDAT transgenic plant, and we see this very large oil droplets. This fluorescence microgram, as well as this is electron microgram. So one phenomenon that during our study we found is really informative is that if we look at the oil level or TAC level in different in leaves at different stage, I mean young, middle-aged, old leaves, we see that TAC only accumulated in the green leaves. It's once leaf become mature or senescent, TAC levels drop dramatically. So this tells us that oil is produced, but it's degraded very rapidly. So this led to our another strategy to increase oil content, blocking TAC turnover, because TAC level is a balance of rate of fatty acid, rate of synthesis and rate of degradation. So we know, again, this is knowledge is from seeds, seeds oil seeds, because a lot of study done in seeds, because seeds is very important for food. We know that studying seeds, there are two players here, important in the oil TAC breakdown. One enzyme we call SDP1. This enzyme is responsible for release fatty acid from TAC, so to generate free fatty acid. Another player here is a transport, fatty acid transport, responsible for transport fatty acid into an organelle we call peroxisome, into a fatty acid, transport fatty acid into peroxisome to enter fatty acid degradation pathway we call beta oxidation pathway. So we wondered whether these two enzymes important in this TAC turnover in TGD mutant. To test this, we use a genetic approach. We knock out STP1 gene in TGD1 mutant background, or we knock out PAX1 gene in TGD1 background. Here is what we're very excited to see that compared with TGD1 mutant, this always increase dramatically. So we also test other other candidate gene. None of this is showing any phenotype when compared with TGD1. Again, this suggests that these two are major players in tech breakdown in leaf. So we measure the oil content. This is what we get. We saw a 150-fold increase to 9% by dry weight. This is the highest level reported in Arabidopsis research. Arabidopsis leaves, in leaf Arabidopsis growing on soil. So, this is how oil droplets look like again. So you disrupt, remove, blocking the tag turnover, you see dramatic increase in size and number for your droplets. This is very big oil droplets. Here is EM picture, electron micrograph. So the plant's biomass is not significantly effect. Here is TGD1 and TGD1 STP1 dominant, TGD1 PAX dominant. The overall height as well as biomass is not very different from TGD1. So I already show you that by blocking the fatty acid turnover, we can get this much of oil. So we ask a question that whether we still can further increase this oil by targeting different gene or pathway. So in search for candidate gene or pathway, a family of protein called oleosin came to our attention. So what's the oleosin? Oleosin of seed-specific protein. That means seed-specific is only expressed in seeds. They are protein that coating seed oil droplets, located only at the surface of oil droplets. It's believed to play an important role in protecting oil droplets from breakdown. So plant leaf do not have this oleosis. And this is how oil droplets in plants leaf look like. And we wonder what happened if we overexpress this oleosin in leaf that do not have this oleosin. It's interesting. So this is what happened. When we overexpress this oleosin in these plants, this plant already have a PTET gene overexpressed. We see a lot of very big oil droplets. And if we overexpress a lot of gene oleosin 1, 2 dramatic phenotype happened. One is that the oil droplets become much more smaller. The second one is that these oil droplets form a cluster installed for this individual oil droplets. This clustering phenotype is more clearly seen using electron microscope. We see a lot of cluster of oil droplets. This is my favorite picture. I show a lot of time in the presentation. Here you see the chloroplast. This oil droplet cluster is even bigger than this chloroplast. And here is a central vacuole. This is a part of plant cells. So when we check the oil content, it's a little bit disappointing. It's not further increase above this 9%. But still, we feel that at least this is the way that they can, compare with this genetic background, you overexpress oleosin, they still increase dramatically. Right now, we are testing whether we can overexpress in this gene other mutant genetic background to see whether we can further increase oil above this level. So plants look happy in this transgenic line, overexpress couple of genes, peanut and oleosin. So in the last part of talk, I will go briefly give some thoughts on our future research direction. So I already showed you that we have identified a mutant that show a fourfold increase in fatty acid synthesis and increase availability of fatty acid for oil synthesis. This pathway remains constant, no change. So when we overexpress this enzyme, along with oleosin, we can get this much of oil, 9% by dry weight. And similarly, if we block the fatty acid oil turnover pathway, disrupt either one of these two genes, we get the same level of triacylglycerol. So, obvious question in the future we wanted to answer is, can oil be further increased? One interest area we want to test in the future is that the interaction between starch and fatty acid synthesis. So, photosynthesis is the source for, only source for sugar, which is used for producing either fatty acid or starch. So one of the obvious questions, can we block starch synthesis to make it more sugar available for fatty acid synthesis, therefore increase in oil-based synthesis? The other area of interest is how fatty acid synthesized regulated at the transcription level. What the mechanism behind this fourfold increase in fatty acid synthesized in a TGD1 mutant. So more importantly, can we produce oil, increase oil in a real crop? Can we transform this knowledge into real crop? Because of very high biomass yield. So even if you can make a small increase in oil content in high biomass crop, it will have a very big impact on the overall oil yield. For example, here, if we can produce 5% of oil or 5% of oil by dry weight in mixed cancers with a very high yield bioenergy crop, the oil yield per acre is equal to canola, major oil seeds crop in the world. If we can make 10% of oil by dry weight in a biomass crop, the oil yield will be double compared with oil seeds canola. So perhaps in the future, we can use this plant oil to make a biodiesel to drive a car in a renewable, sustainable way. So lastly, I would like to acknowledge people who contribute to the work I'm talking about today. So most of data I talk about today is generated by Jin, and Changcheng Xu participated in a radio tracer labeling study. And we have some collaboration project with John York and also Christoph Benning. And John helped us throughout the research and in many different ways, from experiment, writing, to today's presentation. And I cannot see, this thank enough for what John has done for us. And I also want to thank to Bill, Paul, and Changcheng and Huilin for their help, their generosity in sharing chemicals and equipment ideas. And I would like to thank you. And I also want to thank our financial support, which is from Office of Basic Energy Science. And I want to thank you for your attention.
Right, we have time for a few questions. Sean?
Did you try knocking out SDP1 and PXA1 both?
Yeah, both the same oil, the same. Yeah, this they are sitting in the pathway, the linear pathway by the different step. So you knock out both, they very similar to. So the question is, can we, if we knock both SDP1, PAX1, both gene, whether they produce the same phenotype as mutant knockout single. So my answer is that they are similar. Oil, in terms of oil accumulation, they accumulate exact same amount of oil because they are sit in a linear pathway, just 2 steps. So if you knock both, they just produce the same amount of oil.
Yes, down here.
So you mentioned specifically that it was the most oil that was produced when it was grown on soil. Is there a reason? Is there something, another way you could grow it that would produce more oil?
Can you repeat it again?
So you mentioned specifically that it was the most oil that you had produced in Arabidopsis when grown in soil.
Uh-huh.
Is there a different way of growing it that produces more oil?
Yes, yes. So she's asking whether we can produce more oil than the oil we see when plants are growing on soil. The other way, I mean, obvious ways we can grow in plants on agar plates, supplemented with sugar, which is a substrate for oil biosynthesis. If you provide more sugar, they will produce more oil. Probably more oil will higher than the plants we see when growing on soil.
Can I add a comment there? Other people in the literature had grown plants on sugar and faked their data, so he was making the point that these are authentic data, okay? Yes, down here.
Does the extra oil content in the leaves affect the plant's hardiness and insect resistance and that sort of thing?
Yes. So the question is whether extra amount of oil will affect the plant's performance, right? Like insect resistance or other kind of traits, agriculture, agronomical traits. We have one piece of data that, not our data, other people data that if you accumulate more oil in leaf, insects more likely. The insects tend to eat the plants with more oil, and these insects are growing bigger. Then plants feed with, then insects feed with plant without oil. I have this data I originally wanted to show, but I saw it might be too much. I didn't.
Waylon?
Tony, can you tell us a little more about what this oleosin and the code this oil droplet? Is this a protein or just organic molecule?
Yes.
And when you overexpress the gene for this oleosin, do you characterize it and how much this molecule is produced in a leaf? And by producing this, and does that eliminate this sort of developmental stage related to this oil distribution?
So this question is whether we the oleosin, or what's the oleosin, right? Oleosin give more information about the oleosin, whether we overexpress oleosin, how much oleosin protein we can see in leaf. Is that your question? So the oleosin is a structural protein. I will see, based on most of the information. They just located in our oil droplet surface. By doing this, we call coating. They are just coating the whole droplet surface of seeds oil droplets. Seeds because, like Arabidopsis, the seeds is a major organ for storing oil. So seeds oil droplets coated with this oleosin. So oleosin, other than structural roles, I think they don't have other enzymatic functions. So when we overexpress this protein in leaf, when we fuse, I have some nice picture, but I didn't show because of time limitation. So time limits. So when we fuse this oleosin with the GFP, green fluorescence, when we overexpress this fusion protein in leaf, we see this whole droplets become green. It's very nice. Actually, this, oops, it's the wrong. Let me show. This is the one that how when you overexpress oleosin GFP in leaf look like. So you see this oil droplets. You only see this. This is actually oleosin GFP labeled. So when you overexpress oleosin GFP fusion protein, you use this kind of circle. So it means that this oleosin protein is on the surface. And then lots of oleosin proteins expressed based on fluorescence image.
Ben, right at the back.
Changcheng.
Hi.
Two questions. One is a quick clarification. The 10% oil, is that in mature and senescent leaves? And
Yes.
The second part is, do you think it would be possible to have these mutations turn on at the end of the life cycle so that you don't have the oil accumulating until the end?
Yeah, that's.
To try to deal with the herbivore problem.
Yes, the question is 10% is observed at what developmental stage, right? Second question is, can we turn on the mutation at, for example, late stage of development? And the first question, I would see that this is highest, 10% is the highest observed. It's normally low at beginning, young stage. Oil will become higher as leaf become mature, and the highest oil is observed at leaf towards senescence stage. But if you reach complete senescence, the oil will drop, probably they are degraded again. So that second question, whether we can turn on this mutation at a specific stage, that's an interesting question. And in this way, the mutation will not affect the whole biomass production, biomass yield. That's the interest area that we can look at in the future, but we don't have experiment data to data indicate any. I cannot give you any answer yet. Because we haven't done anything.
We have another question down here.
So you showed an application of this as a carbon neutral way to produce energy from the sun. Do you know how it compares in terms of efficiency with other ways to convert solar energy into energy we can use to drive cars or turn on lights?
I don't know the efficiency yet, but this is a very early stage. But from this, at least theoretically, we can see the benefit that we can, because car release, driving car on fuel, they release carbon dioxide. This carbon dioxide can be refixed by plants. This generates a good circles, and we don't have net release of carbon dioxide. And at least the benefit to environment. As far as the efficiency is concerned, I really don't know. Like the photosynthesis efficiency, it's low. Like 2%, I guess, is probably 2% of light to chemical energy conversion power is highest. They're normally below this level. So the efficiency by plant to fix sunlight into a usable energy is very low. And this probably is not my area for research. But it's a very interesting question. A lot of people look at it, how we can improve the efficiency of photosynthesis. That's probably. Oh yeah, all right.
Paul, you have a question?
Another way to ask that question, early on in the talk you talked about percentage of biodiesel from seeds.
Uh-huh.
If all of it went to oil,
Mm-hmm.
it's a very small amount, 4%.
Mm-hmm.
So did you do any calculation, or has anybody done any calculation that would project how much oil could be produced if you were successful in doing this for leaves, how much biomass you would produce, and what percentage of the biodiesel could be produced?
The question is, yes, looking forward. Do I have any idea if we produce this much of oil in vegetative tissue, how much impact this will have on the fossil fuel consumption? The answer is I don't know. I'm sorry.
Well, if you take the numbers from your talk, you said it was about 4%, right? And if you could get tenfold more per plant, per land acre, then that could be 40%, I guess. Just...
Yes, that's probably... See, John know better than me.
Can I call a friend?
Yes.
Okay, let's see. Question here.
Your procedure increases oil production, but at the same time trying to keep the food production about the same. How about the quality of the food? Will this procedure alter the quality of the food?
Yes, the question is whether we produce oil and leaf affect the seeds quality or food quality. That's probably another important question needed to be answered in the future. My answer is again, I don't know, probably John knows, but I'm sorry. So this actually is a very active area. It's a very interesting area, deserves further investigation. And whether we produce this oil in leaf, it will affect the seeds production. Overall biomass, based on all observation in Arabidopsis, look like if we accumulate 10% of oil, the plants still looks happy. But insects make it. Insects eat this high oil accumulation plants. This probably, it has bad impact on food production. But we can separate food and fuel production. We can simply use these plants produce only leaf oil and let the other plant to produce seed oil. That's all possible. You can use the high biomass energy crop to produce oil and leaf, then let the corn, soybean to produce food for us.
I'd like to ask one last question, and that is that Well, I was going to just say that in these plants with 9%, you still had normal seed set, right? So that indicates that you can have 9% of oil in leaves and still have normal seeds. But the question I was going to ask was about Arabidopsis has a very short growth season, you said, and that's why you chose it to work on.
Yes.
Does that work against you in this case? And if you could, in a crop that grew for a lot longer, would you think it would accumulate more leaf oil?
That's a good question. And we need plants growing non-season growing plants to test this. And theoretically, Arabidopsis is a very short life cycle. Like most of the crop, it's much longer. For example, tobacco, from seed germination to seeds production probably take half a year or something. So if we can produce similar, achieve similar rate of oil accumulation in tobacco as we observed in Arabidopsis, the overall yield, oil yield, oil content is likely will be much higher in tobacco because they have much longer growing season.
All right. Well, I think thanks very much for answering all those questions, and I'd like to ask everyone to join me in thanking Changcheng again for a great lecture.
Thank you.